Hi, thank you again, Alexandra, for your kind introduction. And hi, everybody. Today, I'm going to talk about genotype imputation using the UK Biobank whole genome sequencing data. And interestingly, this talk follows pretty well the previous virtual meetup, where we have seen that our haplotype phasing methods are now scaling to the current large amount of data that UK Biobank has been made available. And we also follow up on that, showing how these very accurate haplotypes actually can improve genotype imputation and rare variants. And I mentioned high coverage whole genome sequencing, as the UK Biobank has made available 200,000 UK Biobank sequencing, and it will extend this, and by the end of the year will be 500,000. So every single sample will be available. And this produce, as we know, 30x high coverage genomes are used as a standard to produce very accurate genotypes. The major drawback, however, of local high coverage whole genome sequencing is its cost. Now it's getting a bit lower. In the last year, probably is around $200 per sample, but until a few years ago, it was really prohibitive. So in the context of genome-wide association studies, major biobanks have been using another technology called SNP array. And SNP array actually focused only on specific positions in the genome, approximately 1 million variants. And then a bioinformatics tool called Genotype Imputation is used to increase the resolution of the data. I will explain what's genotype imputation in a minute. But then with this imputed data now, it's possible to run a genome-wide association study and hopefully boost the signal with imputation. This is how SNP array data looks like. And as you can see, only few of the variants are actually typed on the SNP array. We have missing data all over when the variant is not typed on the SNP array. And as I said, imputation is used to infer statistically the genotypes that are missing. I also would like to add that there are also other technologies that can be used that are much cheaper than high coverage whole genome sequencing. One of them is low coverage whole genome sequencing. And in this context of this talk, I'm talking about low coverage, approximately like 1x coverage, or something that goes from 0.1x to 4x coverage. And even in this case, we have a much reduced coverage. So we have a lot of missing data, and the genotypes need to be refined with genotype imputation as well. So very briefly, to illustrate how genotype imputation works, I use this very nice illustration from Marchini and Awi paper from 2010. On the bottom left of the slide, you see many samples. Each row represents a sample, SNP array data, so full of missing data. And of course, we could run a genome-wide association study. And if we look at the Manhattan plot and log-time p-value, likely we will get a signal that is not very clear because of the missing pattern of the SNP array data. So genotype imputation is used here, and it's not magic. It's using a large reference panel of haplotypes, so an external data set, whole genome sequencing data. This can be 1,000 genomes or the HRC reference consortium. Or in the reminder of the talk, we will also the UK Biobank reference panel to perform genotype imputation. And imputation works in two different steps. First of all, we get, for each single sample, we get the two haplotypes. So we do what is called pre-phasing. So we get the two chromosomes of the sample. And then these samples are now can be modeled as independently as a mosaic of haplotypes in the reference panel. And this is what a genotype imputation is modeling each of the haplotypes, and the imputation is simply a probabilistic copy of this mosaic. Eventually, we combine the two haplotypes together, and we get a probabilistic value for each of the genotypes. Now, this is not very clear here, but the amount of markers that are imputed is much bigger, approximately 100 times bigger than the amount of markers that you get on the SNP array. And now, since you have many more variants, although imputed, if you run a genome-wide association study, you will be able to boost your signal. And we can see an example of this from the UK Biobank paper from 2016. where here I show genome-wide association study on standing height for 350,000 samples. On the top, only using SNP array position, and at the bottom, using imputed data. So we go from 800,000 SNPs genome-wide to 40 million SNPs genome-wide. And as you can see, the signal at the bottom is much more clear, is much more in high resolution, and we are able to detail much better the peaks, capture new associations, and overall, I even capture, it is extremely useful for fine mapping because it's likely that the causal variant might be in the imputed data instead of the genotype. So this is why genotype imputation is standard practice in the genome-wide association studies starting from SNP array. However, there are several methods for genotype imputation, but method-wise, these methods these days really converge to the same haplotype, to the same mosaic haplotype model I described you before. It's an hidden Markov model. It's called Lee and Stevens hidden Markov model. All the major methods use the same model. And what the difference between the methods, it's actually on scalability on very large reference panels. So it's mainly computational improvements of the new methods. And indeed, during my PhD, I've been working on a method called Impute 5, so the fifth version of this Impute method that is really designed to create a custom reference panel, a much smaller reference panel for each haplotype in the reference panel. And we do this, we are able to select just the very important haplotypes using a data structure called positional Borzoi transform. By doing this, we are able to leverage much bigger reference panels, and it's motivated by the fact that there is an exponential growth in the number of reference haplotypes that are being made available and the new whole genome sequencing data that is made available. And of course, performing genotype imputation, the reference panel is a key feature. Larger is the reference panel, better will be your imputation. So it's not really much in the method in itself, it's about the scaling. This Impute 5 method has been recently used by Sinan Shi from the University of Oxford to perform genotype imputation of 500 UK Biobank samples using Genomics England as a reference panel. And Genomics England is a UK-based dataset, is relatively not easily accessible like the UK Biobank, but it contains approximately 100,000 whole genome sequencing samples. And we imputed all the 500 samples from the UK Biobank SNP array data, and the data is already available on the UK BRAP. So if you're using imputed data, I recommend to use this dataset. However, the UK Biobank has also made available 200,000 whole genome sequencing samples. And of course, this is an amazing opportunity to use this data as an actual reference panel for imputation. And for this reason, we work pretty hard to extend our haplotype phasing model to make it scalable and to improve the accuracy of rare variant haplotypes, rare variants. And this method is called ShapeIt5. It has been released late last year. And we were able to verify the impact of our haplotypes using SNP array imputation. And indeed, it's because we are in a very interesting scenario using the UK Biobank platform. We have SNP array data for the 200,000 samples, but also 30x high coverage whole genome sequencing data for the same sample. So we can phase the full high coverage whole genome sequencing data, extract, for example, 1,000 samples, impute using from the SNP array data, using the other samples as a reference panel, and then use 30x data for these 1,000 samples as a validation. So we are really able to benchmark how good are our haplotypes using using genotype imputation as a procedure to evaluate it, as we don't have many trios in the UK Biobank. And this is what an imputation plot looks like. And on the x-axis, you see minor allele count in the reference panel. In this case, we go up to 200,000 reference panel samples. And on the y-axis, we have aggregate R-square. It's just a measure of imputation accuracy. So it's a measure of the accuracy of our imputed calls compared to the 30x data that we have available on the platform, that we treat them as a ground truth. So as you can see, the variants are binned according to the minor allele frequency, and we have this kind of very, very common shape in genotype imputation. And here we do imputation using the first release of 150,000 samples, and additionally, the additional 50,000 samples that have been released. And as you can see, we obtain really high quality genotype imputation, reaching an R-square of 0.8 for variants that are present with a minor allele count of 11 to 20 in the reference panel. To do this, we actually work a bit on improving our imputation tool called Impute5, and we will be able to release a new version of this software relatively soon. We estimated that the whole genome cost to impute one single sample on the UK Biobank route using this setting would be approximately 0.3 cents per sample. So it's extremely cheap. It will be able to scale to even almost million of samples for just a few thousand UK pounds. So this is what I wanted to tell you about SNP array imputation, the latest development we did. And I would like now to move towards another field, another type of technology, that is low coverage whole genome sequencing. Low coverage whole genome sequencing, as I told you, here I refer to low coverage as 1x data approximately, is not new. It has been introduced approximately in 2012 by a very seminal paper by Bogdan Pasianic, that he was able to evaluate actually off-target reads in whole exome sequencing data, treating it as a low coverage experiment against SNP array, and showing that this low coverage they will get from the off-targets in the whole exome sequencing data will actually increase the power of genome-wide association studies compared to the available SNP arrays. These results have been replicated by other groups. However, there are not many methods many data sets that have been using low coverage whole genome sequencing. And one of the main reasons here is that it's mainly the computational bottleneck. When I explained to you genotype imputation very briefly, I mentioned this two-step approach, pre-phasing and then imputation. However, in the context of low coverage whole genome sequencing, we are not able to separate these two steps because we don't have a set of very high quality calls we can then phase, but phasing and imputation are kind of built in together in the model. The four methods tend to be quadratic in the as a cost in the size of the reference panel. And when I moved, just to give you a concrete example, when I started doing some imputation on low coverage sequencing data, methods were in 2019 were not even able to scale on the 1,000 genomes reference panel. They were really struggling to use an haplotype reference panel derived from low coverage data for 2,500 samples. So it was extremely expensive. For this reason, we introduced a method called GLIMS. And the key feature of GLIMS is the fact that it's able to perform haplotype sampling in linear time instead of quadratic time. So basically, instead of doing one very expensive run of deployed imputation, we do two much quicker runs of linear haploid imputation. So it's linear time instead of quadratic time. Additionally, we also borrowed some of the new innovation we use for SNP array imputation, such as they use the PBWT data structure. So we will also collapse the data set to very informative state. And this is able to perform imputation somewhat constant in the number of reference samples, but linearly in the number of variants in the reference panel. And just to show how low coverage whole genome sequencing imputation performs against SNP array data, I'm showing these plots from the GLIMS1 paper. So here you have two different low coverage experiments, 0.5x imputed data and 1x imputed data with 2 shades of blue, and 2 different SNP arrays. One is the UK Biobank Axiom array, and the other one is the GSA array. So widely used SNP arrays. And as you can see, pretty much both technologies perform the same at common variants. They pretty much have the same accuracy. However, low coverage whole genome sequencing is more accurate at rare variants. And this makes sense also because we have much more information on low coverage whole genome sequencing compared to SNP array data, where we have only missing data. Additionally, on the other side, I show that by increasing the reference panel size, we keep increasing imputation accuracy, even in the case of low coverage whole genome sequencing. Therefore, we are extremely motivated to use the newly phased UK Biobank haplotype reference panel to see how low coverage imputation will perform with data of that size. However, this is not so simple. We needed to take the glimpse model and add many methodological improvements to scale on data containing 150,000 samples and approximately 600 million variants, sorry. So it's extremely large. It's very complicated to make a method that is able to scale on this size. However, we exploited the sparsity of the structure. So the main component in the GLIMS2 model that we proposed and released at the end of last year is really using a sparse reference panel representation. And indeed, most of the variants that very big cohorts of whole genome sequencing data bring are extremely rare. So by exploiting this sparsity, we are able to perform a sparse probability selection and sparse imputation of variance, basically not wasting time doing useless computation. The method is available on bioRxiv, and we release the preprint, as I told you. So how does the UK Biobank reference panel perform on low coverage whole genome sequencing data? And what I'm showing here is actually 3 different reference panels. So we took 100 samples at 1x coverage, and we imputed them using the 1000 genome reference panel, the HRC reference panel, and the UK Biobank reference panel. Of course, given the size of the reference panel, it's possible to go to much lower minor allele frequency with the UK Biobank reference panel. It's much bigger, so we can go to extremely low minor allele frequencies. But overall, it's striking that the performance of the UK Biobank reference panel achieves compared to the previous 2 reference panels that were available. And this is using GLIMS version 1, but when we bring GLIMS version 2, we are able to even further improve the accuracy at rare variants, especially for the UK Biobank reference panel. And this is possible because now we have such a sparse model to represent the data that we can relax some computational constraints we had on the GLIMS1 model. We are able to use many more states, and this will improve the accuracy at extremely rare variants. Importantly, these improvements are also achieved with a fraction of the running time. And you can see it here. For the UK Biobank reference panel, GLIMS version 1 will cost more than £1 per sample whole genome, while GLIMS version 2 keeps the cost under 10 cents. And this is crucial because we need to keep the cost extremely low if we want low coverage whole genome sequencing to be widely adopted. This has to be a fraction of the cost that we will pay for the sequencing. So this is using the UK Biobank wrap, and yeah, so given also the fact that we use this reference panel representation, we are able to go to very small, much smaller instances, so to keep the memory very compact. Importantly, also, we benchmark different coverage configurations from 0.1x data, 0.25x data, up to 4x data. And of course, the imputation accuracy increases with the coverage. And we also added here in red the UK Biobank Axiom array imputed with the same reference panel, of course. Therefore, in this case, we have also a very interesting approach, because the UK Biobank axiom array has been specifically designed for the UK Biobank data. Therefore, it's the most ideal scenario for SNP arrays, because they've been really selecting variants to improve genotype imputation for this specific population. And what we actually note here is that the SNP array data performs very similarly to 0.25x imputed data and at common variants, but it actually 0.25x data seems to be better at rare variant, similarly to what I noted before. And interestingly, also, we can go to a coverage as low as 0.1x, which is literally no data, and achieve an imputation accuracy of 0.9 of R squared at common variants and 0.8 at somewhat rare variants. Therefore, we can, even 0.1x can bring very interesting results in the case of genome-wide association studies. And indeed, we further explored this by we took these 10,000 samples we have been imputing, and we ran it genome-wide, and since we have UK Biobank phenotypes, we can now run GWAS. And what I show here is different locus zooms for different technologies. So there are 3 different locus zooms as rows in the figure, and technologies are columns. So axiom array data, 0.1x data imputed, 0.25x data, and 1x data. So also we have the 30x data that we can also use for the GWAS, and we did that, and we note, as I don't know if you can see it, but as a dotted line, as a dashed line, you can see the lead SNP for the location of the lead SNP for the 30x data. And overall, this locus zoom look remarkably similar, almost indistinguishable by high, except maybe for the 0.1x data that you can see there is a small drop of compared to the 30x. And this is due to the inaccurate genotyping we will get from the imputation. However, overall it works really well, even though sometimes we lose the actual significant association. So this is just visually, and it seems to be working extremely well, but we can also do it quantitatively. And in particular, we can take all the betas and the p-values for the 30x data and each of these experiments, and run a correlation between each of these experiments and the 30x data to see how it is the reduction of power that we get from imputed SNP array data or imputed low coverage compared to the 30x. And as you see, this kind of plot is remarkably similar to what we showed before in terms of validation. And indeed, there is a direct effect of the accuracy of the imputation of the genotyping to genome-wide association power. And again, 0.25x data is remarkably similar to the SNP array data, as I told you before. And there is a reduction of power for 0.1x data, but still, if we look, for example, at the betas at 0.1x data, we reach an R squared of 0.92. And this is an important quantity because the betas are used for the polygenic risk score prediction. Therefore, what is the message here that even 0.1x data is actually extremely useful and probably can be used fairly well to perform polygenic risk score prediction. Finally, I would like to say that what we show so far, it's only on British samples. So the idealized scenario for using this reference panel. But of course, we were very interested to see where the UK Biobank, how the UK Biobank reference panel performs compared to other ancestries that are other human ancestries. And for this reason, we took 276 samples from the Simon's Genome Diversity Project. We downsampled to 1x, so it's a project that it's available, high coverage whole genome sequencing data, and we downsampled to 1x and perform imputation using both the UK Biobank reference panel and 1,000 genomes reference panel that contains population from all over in the world, but it's much smaller. And overall, what we see is somewhat what we expected. We see that UK Biobank reference panel performs better than the 1,000 genome reference panels for European samples, here denoted in red, especially for Northern European samples. Of course, English and Scandinavian ancestry. And the 1,000 genome reference panels actually perform better for populations that are not well represented in the UK Biobank, but are represented in 1,000 genomes. So, for example, South American populations and East Asian populations, for example, Japanese population is one of those that gains more with 1,000 genomes. So overall, this is just a note saying that the UK Biobank reference panel is a game changer for many European populations. And we are really looking forward to apply the method to the 500,000 whole genome sequencing samples when this will be available. But of course, there is also a need in the field to produce very much more diverse reference panels, to produce unbiased genotype imputation for different populations across the world. So this concludes my talk. I would like to thank the group of Olivier Delano at the University of Lausanne, where I worked until a couple of months ago. And in particular, I would like to mention the work of Diogo Ribeiro and Robert Hofmeister that have been really working extremely hard for the shape-it file analysis. And I would like to thank the UK Biobank participants and to you for listening. And I'm open to any question.
So for the slide number 12 that you try to show the improvement for the 150K compared to the 200K. Yeah. I just wonder, when you get more data from the biobank here, do you see the shift in terms of like how is the variant distribution in terms of their mononuclear frequency count? Because let's say you might run into the same variants again in that population, right? But then at the same time, you might run into the new variants again. So I just wonder, I just try to expect that when we have the 500K, expect improvement came from that graph shipping up, or also like shipping to the right as well.
Right. That is a very good point. What I did there is probably, maybe I can go to the plot quickly. I think you're referring to this plot.
Yes.
What I'm doing here is indeed not comparing the same variants. Ideally, we would like to, I could have looked at the same variant in the two data sets, and you would have seen, the actual improvement using one or the other reference panel. What I'm just showing here is very simple. Like if you see, in this specific case, this doesn't go, of course, to the 150K. So it's been, it's basically I'm trying to overimpose the two to make them on the same scale. So actually, many of the very common variants that are in the last bin here are probably in this bin, right? And this can probably be shifted. So likely this plot is actually underestimating a bit the increase of performance of the 200,000 reference panel. But yeah, I agree with you that probably a very unbiased way will be just to focus on the same variant and like on the intersection and just look at the differences between the two, and you will see a boost given by the 50,000 more samples more is a very big amount. And this is because genotype imputation works identifying very large identity by descent segments, and of course having 50,000 more samples, we are more able to find a very recent common ancestor, and this is the motivation behind the fact that increasing reference panels, we are able to improve imputation accuracy.
So can you provide a rough estimate, let's say in this case, compare between the 200K and 150K, right? But in terms of the overall accuracy improvement, because as you mentioned that there is a shift in both in terms of at each point on the x-axis here is shipping up, but also you're going to see like more count for the same variant that you already saw before. So it's shift to the right as well. So do you have rough estimate like how much improvement of this in terms of overall accuracy?
All right. Well, I believe, the main differences will be at rare variants, first of all. We see actually something happening also common variants, but I'm expecting the majority of the difference to be at rare variants because 150,000 samples are enough to capture very well common variants. They're easy to impute. In terms of amount of, I think the return of increasing reference panel size is somewhat logarithmic. So by increasing the number of sequences, we should not expect like increasing the 2 times the size of the reference panel. We shouldn't expect a 2 times increase in accuracy. But definitely there is a boost here that we have 50,000 samples more, which is a considerable more than 50%, slightly more than, yeah. So it's a lot of data, and so we will bring some additional boost. Quantifying it, like, I mean, we cannot quantify it using, for example, the R-square, and we will need to look at the same variant in the intersection. And we didn't do that, yeah.
Thank you.
I have kind of a, I guess, a more general question, but I really enjoyed your talk. Thank you so much. I think it's really exciting. I guess as like a fellow methods person, it's really exciting to see these methods. And so kind of with the low coverage imputation of GLIMS2 and your phasing SHAPE-IT 5, I'm just curious how you kind of envision researchers using these and what they're able to accomplish with these.
Cool. Yeah, I'm pretty sure, like, for example, we really focus in this talk on having, for example, for imputation of low coverage, very, flat coverage 1x across whole genome. I believe there are opportunities there to maybe extend this work to, for example, whole exome sequencing, maybe plus some more coverage, low coverage. This is some work that I know it has been is carried on as a potential application. The incredible phasing we are reaching now on short read sequencing data, I'm pretty sure can also be used to somewhat perform imputation of maybe structural variants that I haven't mentioned here. Here the focus is mainly on short variants. So we are really reaching a phase in genetics where we really are probably capping up in terms of in some population in terms of collection of haplotype diversity, and this allows really to reduce the additional sequencing that we need. So given, for example, the UK Biobank reference panel, I don't think it's always strictly necessary to perform 30x sequencing anymore. We can reduce drastically the coverage because most of it will be imputed fairly well. And for most applications, it will be more than enough.
Thanks. Yeah, no, it's really exciting. I think especially with seeing I guess these new methods, just kind of seeing how far everything has come and what you're able to now accomplish with all of it. So thank you so much for sharing.
Thank you for giving me the opportunity to present my work. We have one question in the chat room before we go into the breakout from Saliha. The question is, actually we have 2 questions now. How accurate? Actually, Simone, can you see the question in the?
Yes, yeah.
Right, perfect.
Seeing there is a message. Okay, so perfect. How accurate do you think is the detection of compound heterozygosity detection of rare variants and singletons? That's very. I didn't mention this part of the analysis, but in the SHAPE-IT 5 preprint, we also look. Given now that we have very accurate haplotype phasing, we look at we screen for compound heterozygosity using the whole exome sequencing data plus NIP array. So we are able to detect some gene that actually has a compound heterozygous. And the question is about how accurate do you think that is compared to standard studies that actually work on trios to detect compound heterozygosity. And this is a good question. I feel like, of course, like one of the things that the question mentioned is accuracy at singletons. And indeed, singletons phasing, it's usually, we are not able to perform phasing at singletons. However, with ShapeIt5, we somewhat have a method that is able to detect some, to actually move from the 50-50 phasing to something a bit better. And we evaluate in the paper the compound heterozygosity also with and without singletons. So it's a good point. Of course, in terms of accuracy, it's of course a matter of how the the thing is that we rarely have, like, since given this amazing opportunity to actually look at the data and have these very accurate haplotypes, we can now start thinking about compound heterozygous. I feel like there is, of course, there are some mistakes in using population-based phasing, but overall, I feel pretty confident that the amazing phasing we are reaching, and I've just shown you the results that we really have incredibly good imputation as well at extremely rare variants. For that reason, we probably are able to capture most of the compound heterozygous events. Is there maybe another question? Yes, so Ben is asking, we really capture haplotypes for white British folks, right? What have a long way to go with most other populations? How would you strategize sequencing for other more diverse populations? How can we leverage UK Biobank to kick off that? That's very interesting, especially given the very large haplotype diversity of African population. It's extremely useful and to start thinking on how we are able to capture that. And there is this very interesting paper by Alicia Martin from a couple of years ago that she shows that she actually uses low coverage whole genome sequencing plus imputation to start detecting to analyze many African samples. It's in collaboration from several African researchers. So I believe actually that this work on low coverage whole genome sequencing can be instrumental as a first step towards that, because it's possible to envision that we will do like stepwise, like we start with some lower coverages for several populations that are somewhat isolates or have a very different, very diverse haplotype diversity, and then we will then bring high coverage for genome sequencing, similar to what we did for the UK Biobank resource. So it's somewhat, I would think that, you know, this work on low coverage, moderate coverage such as 4x or 8x can be a first step to capture and detect haplotype diversity, maybe in African populations or population isolates.